Good morning, everyone, and thank you for being here today. I am pleased to welcome you to this presentation on out-of-specification, or OOS, results. My name is Dr. S.K. Singh, and I hold a PhD in this field. Today, we will explore a scientific, structured, and compliant approach to investigating OOS results. Over the course of this session, we'll cover the key steps involved in conducting effective investigations, ensuring that our methods are both thorough and compliant with regulatory standards. I look forward to sharing insights and best practices that can help us in managing OOS situations confidently and effectively. Let's get started. Let's take a closer look at what applies to QC laboratories, whether we're testing raw materials, in-process samples, finished products, or stability batches. These standards are based on current expectations from regulatory bodies like the US FDA, EMA, MHRA, WHO, and CDSCO. The key focus here is on the entire process, from detection of an out-of-specification result to investigating the root cause, making a decision, implementing CAPA, and thoroughly documenting everything. Now, the core concept here is the OOS, or out-of-specification. Essentially, an OOS result is any test outcome that falls outside the established acceptance criteria or limits. These limits are defined in various places, such as the regulatory filings, pharmacopoeias like USB, EP, or IP, or our internal approved specifications. Understanding what constitutes an OOS is fundamental because it triggers the entire investigation process and ensures we maintain product quality and compliance. So, in essence, recognizing and properly managing OOS results is critical to ensuring our products meet all safety and efficacy standards, and it helps us stay aligned with regulatory expectations. This is a vital part of our overall quality assurance system. This slide outlines the scope of our APLITA considerations, which include release and stability testing across various stages. We look at raw materials, intermediates, and finished products to ensure quality at every step. Both microbiology and chemistry tests are involved here because each provides critical insights into product safety and efficacy. We also need to consider results that are out of trend, or OOT, which are not necessarily out of specification, but they can still trigger an investigation. This is important because even trends that deviate slightly might signal underlying issues that need addressing before product release. Now, when we talk about out-of-specification results, or OOS, these are numerical results that fall outside the established upper or lower limits defined by our specifications. These limits are set based on regulatory and internal standards to ensure consistent quality. A test result might also fail to meet key quality attributes like identity, potency, purity, assay, impurities, microbial limits, or other important metrics. If any of these are not within acceptable ranges, it could compromise the product's safety or effectiveness. It's also crucial to compare our results against predetermined and approved standards, which are documented in various references like the Product Registration Dossier, whether it's an ANDA, NDA, or MAA, our internal quality standards, ICH guidelines like Q6A or Q6B, and compendial monographs. Any deviation from these can be a red flag. Now you might ask, what exactly is OOS? Well, OOS stands for out of specification, which means a result has fallen outside the predefined limits. Recognizing OOS results promptly is essential because they often require thorough investigation to determine root causes and ensure that only quality products reach the market. When it comes to out-of-specification, or OOS, test results, regulatory guidance is quite comprehensive. In the U.S., the FDA provides specific guidance for the industry, emphasizing the importance of thorough and scientifically justified investigations. They stress that investigations should be timely and free from bias to ensure the integrity of the quality process. Moving to Europe, the EMA's guidelines are embedded within their GMP regulations, specifically Parts I and II, along with Annex 1 for sterile products and Annex 15, which covers qualification and validation. Their focus is on maintaining high standards through rigorous quality control practices. Globally, organizations like WHO, PICS, and ICH have established key guidelines. For instance, Q6A and Q6B specify requirements for drug product and API specifications, while Q7 covers good manufacturing practices for APIs. Q9 introduces risk management principles, and Q10 emphasizes a robust pharmaceutical quality system. These guidelines collectively underpin a consistent approach to handling OOS results. National regulators, such as CDSCO in India, tend to align their expectations with WHO, FDA, and EMA standards. They expect companies to have well-defined, robust SOPs and investigation practices for OOS results. The common theme across all these guidelines is clear. Investigations must be thorough, conducted promptly, unbiased, and scientifically sound. This ensures that any deviations are properly understood and managed, ultimately safeguarding product quality and patient safety. Now let's take a closer look at the different types of out-of-specification, or OOS, results we might encounter. First, we have true OOS, which means the sample or product truly failed to meet the established specifications. This is a clear indication that something went wrong with the process or the material. Next, we have laboratory error or assignable cause. These are errors that can be traced back to specific factors like the analyst, the equipment, reagents, or even calculation mistakes. It's important to identify and document these causes because they can often be corrected and prevent future issues. Then there's the atypical, aberrant, or outlier result. These are statistical anomalies, values that are unusual but still fall within the official specifications. They don't necessarily mean there's a failure, but they do warrant attention and further investigation. Finally, we have out-of-trend, or OOT results. These results are within the specified limits, but are abnormal compared to historical data. This is especially significant in stability studies, where trends over time can reveal subtle issues that might not be obvious from a single test result. It's crucial to remember that each of these types requires a documented evaluation. We should never discard data without a solid scientific justification, as every result can provide valuable insights into our processes. When approaching results, we should assume they are potentially valid until we have clear evidence to prove otherwise. It's important that we avoid the trap of testing into compliance, which means we shouldn't keep testing repeatedly just to get a passing result. Instead, our focus should be on conducting investigations that are timely, that is, started immediately when something is detected. These investigations must also be unbiased and independent. That means supervisory or QA oversight should be involved to ensure objectivity. Additionally, all investigations need to be well documented, traceable and reconstructible, so that every step can be reviewed later if needed. Most importantly, our final conclusions should be scientifically sound and driven by the data. When it comes to out-of-specification investigations, or OOS investigations, these principles are especially critical. Following these regulatory principles helps us maintain integrity and confidence in our results, ensuring we are making decisions based on solid, unbiased science. Let's start by verifying the sample identity, making sure the label and batch number are correct, and reviewing the dilution schemes used. It's also essential to double-check the analytical method we're following and ensure we're referencing the current version of the SOP to maintain consistency and accuracy. Next, we need to check the instrument's status, confirm that it's properly calibrated, and review the maintenance logs to ensure everything's up to date. Examining the chromatograms, paying attention to integration parameters and system suitability is crucial to verify the system's performance. We also want to review the reagents and standards, making sure their validity is current and that they are stored and prepared according to records. Don't forget to scrutinize the calculations, units, significant figures, and rounding to avoid any errors that might impact our results. If there's any doubt about the sample or data, and it's justified, we should consider performing a reinjection or a repreparation of the solution, but only under controlled and well-documented conditions. The goal here is clear. Either we identify an assignable lab error that explains the issue, or if we find no lab error, we move forward to phase II, which is the full-scale investigation. This initial step is fundamental because it helps us systematically rule out common issues before diving deeper into the investigation, ensuring our process remains rigorous and reliable. Now let's talk about the cross-functional team involved in this process. It includes QC, QA, production, engineering, and regulatory. These groups come together to evaluate the situation comprehensively. We start by reviewing batch manufacturing and packaging records to identify any inconsistencies or issues. Then, we look into process deviations, alarms, excursions, and environmental conditions that might have impacted the batch. Cleaning processes and cross-contamination risks are also assessed carefully, since these can affect product safety and quality. We examine in-process controls, other test results, and stability data to get a full picture of the batch's performance. Next, we evaluate the impact of these findings on other batches, considering their strengths and the markets they serve. We also look at the product that is currently on stability testing or already in the market to understand the broader implications. Based on this assessment, a critical decision must be made regarding the batch disposition. This could mean rejecting the batch, reworking or reprocessing it, or accepting it with strong justification if the data supports that choice. Finally, we determine if there's a need for a product recall or field alert, in line with local regulations, to ensure patient safety and regulatory compliance. When we move into phase II, which is the full-scale investigation, either manufacturing or system-related, we need to dive even deeper. This involves a thorough, systematic investigation to identify root causes and implement corrective actions. It's about ensuring that similar issues won't happen again and maintaining the integrity of our products and processes. Let's talk about retesting first. Retesting is only allowed when there's a solid scientific reason to do so, such as when there's doubt about the solution preparation. It's important to remember that this process must be predefined in the SOP, including the number of retests, the criteria for deciding if a retest is needed, and the overall approach. Moving on to resampling, this is only appropriate if there's a suspicion that the original sample's integrity has been compromised, perhaps due to a mix-up or handling error. When resampling, the sample must accurately represent the same batch and lot, and it should be uniform across the sample. Now, regarding averaging, it's crucial to understand that averaging is not meant to hide an out-of-specification result. In other words, the mean cannot override a confirmed failing value. However, in certain cases, such as testing for uniformity of dosage units, averaging can be used if the method allows it. Finally, all these decisions, whether to retest, resample, or average, must strictly comply with the regulatory specifications filed and relevant guidance. This ensures that our testing strategies are consistent, justified, and compliant at all times. On this slide, I want to emphasize the importance of having a clear out-of-specification, or OOS, standard operating procedure. This SOP should explicitly define roles and responsibilities so everyone involved knows what their part is throughout the investigation process. It should also outline a step-by-step approach to investigating OOS results, ensuring consistency and thoroughness. Additionally, including decision trees for retesting, resampling, or determining batch disposition helps streamline decision-making and reduces ambiguity. Maintaining comprehensive investigation reports is crucial. These should include the problem statement, a detailed chronology of events, relevant data, identified root causes, and a risk assessment to understand potential impacts. Tracking trends is another key aspect. By analyzing OOS or out-of-trend data across different parameters, such as product, method, equipment, analyst, or shift, we can identify recurring issues and systemic gaps in our processes. This proactive approach allows us to implement targeted solutions. Speaking of solutions, implementing effective corrective and preventive actions, or CAPA, is vital. This might involve revising or validating methods, providing additional training, requalifying staff, optimizing processes, or upgrading equipment. The goal is to address the root causes effectively. Equally important is verifying the effectiveness of these CAPAs through follow-up checks, ensuring that the issues don't recur. Finally, I want to highlight that thorough documentation, trending analysis, and CAPA follow-up are essential to maintaining quality and continuous improvement. These practices help us not only resolve individual issues, but also strengthen our overall system to prevent future occurrences. Let's take a closer look at the OOS investigation workflow. This slide outlines the key steps we follow when investigating out-of-specification results. First, when an OOS result is identified, the process begins with a thorough review of the test data and methodology to ensure there were no errors or deviations. Next, we typically perform a retest or reanalysis to confirm whether the result is consistent or an anomaly. If the retest confirms the OOS, we then proceed with an in-depth investigation to determine the root cause, which could be related to sampling, method performance, or equipment issues. Throughout this process, documentation is critical to maintain traceability and ensure compliance with regulatory standards. The ultimate goal here is to resolve the issue efficiently while ensuring product quality and patient safety. Connecting this workflow to our overall theme, it emphasizes the importance of a systematic, disciplined approach to quality control, helping us prevent recurrence and maintain trust in our products. OOS, or out of specification, is a critical GMP signal that goes beyond just being a lab issue. It's an indicator that something might be affecting the quality of our product or process, so we need to take it seriously. When we encounter an OOS result, it's essential to follow a structured, stepwise investigation. This means systematically examining every aspect of the testing and manufacturing process to identify the root cause. It's also crucial to ensure our investigation aligns with current regulatory guidance and our regulatory filings. Staying compliant not only keeps us in good standing, but also reinforces the integrity of our data. Throughout this process, we must prioritize data integrity, transparency, and traceability. Every step, every finding needs to be documented clearly and accurately. Using OOS and OOT trends is another key point. These trends can give us valuable insights, helping us continuously improve our process robustness and, ultimately, our product quality. When we have a strong OOS management system in place, it not only boosts regulatory trust, but also enhances patient safety, because quality and safety are always our top priorities. So, in summary, having a solid approach to OOS is fundamental. It's about more than just fixing a lab result. It's about maintaining the integrity of our entire quality system and ensuring we deliver safe, effective products to patients. For further support, please contact us. If you have any questions or need additional assistance, don't hesitate to reach out. Thank you very much for your time and attention today. We're here to help you succeed, so please feel free to get in touch whenever you need.